Hello, and thank you for joining the webinar. During this education session, we will discuss the actions bioMérieux has taken and continues to take to develop education tools and resources for our customers. My name is Lauren Hunt. I'm an infectious disease pharmacist by training and a medical science liaison here at bioMérieux. I am one of the many members of the Breakpoints task force. This task force is a multidisciplinary team that has a passion for halting antimicrobial resistance, updating breakpoints, and supporting the labs that we serve. I do have to disclose that I am a medical science liaison for bioMérieux, and I do sit on the medical affairs side. In this webinar, it's number 2 of 4 in this education series. I hope you were on for Dr. Prinzi's presentation just before this. If not, I highly recommend going back to listen to it. And while these presentations build on one another, they can absolutely be listened to independently. At a high level, today the topics we're going to cover include a review of our company's commitment to public health, an understanding of the processes bioMérieux goes through to submit new breakpoint requests to the FDA. We'll also discuss the toolkit and all the wonderful things that are in it. And then we'll also share some external resources for our sites. And our objectives are simply an extension of the agenda that I just described. So I am a person who likes to start with the why. And when it comes to breakpoints, there are a lot of reasons it deserves our attention. Antimicrobial resistance is a global issue, one that doesn't understand or respect borders. In order for us to be part of the conversation, to be part of the solution, we must first be able to detect and interpret resistance in order to respond to it. Herein lies a great challenge. Breakpoints are a moving target. The interpretation of minimum inhibitory concentrations via breakpoints evolves constantly, and that's in response to emerging resistance mechanisms, an improved understanding of our pharmacokinetics and pharmacodynamics and how the drug is working on the bug and inside our patients, and new clinical data that says that breakpoints might not be at the right threshold and prompt an adjustment based on clinical outcomes. The revolving door of updating breakpoints presents a challenge for labs, particularly those that might have limited resources such as time or personnel and expertise. However, it's important to recognize that labs that do not update breakpoints not only jeopardize safe and effective patient care for the patients in front of them, it also impedes global efforts to attack antimicrobial resistance. bioMérieux is a global leader in diagnostics, and because of that, we have a duty to support our customers through this journey. Our company vision comes from that of the Mérieux family, to make the world a healthier place. Public health is a common thread through everything that we do here. That includes patient care, both on the acute side and on the outpatient side. It includes veterinary medicine. It includes industry, where we are looking for the safety of cosmetics and food products and pharmaceuticals. And at the end of everything that we do, all of those practices are people. They're the patients we serve through our laboratories. In the case of breakpoints, there was a key publication recently by Patricia Sibner and colleagues. And it showed that depending on the bug-drug combination, labs were using obsolete breakpoints in 40 to 70% of the time for certain bug-drug combinations. So after an initial reaction, this became our call to action. We formed a task force at bioMérieux. There were some like-minded folks that found one another and then continued to bring more into this task force. And you can see that we started back in January of last year, really just aligning some of our key stakeholders. Then at the end of May, beginning of June, we all got together and had our first kickoff meeting. And since then, we've been developing tools and resources, education, putting together a microsite for our customers, and then all of that to disseminate through a communication plan. In November and December, we provided a good bit of education for our internal teams because we had so many questions coming in for customers. And once we finally had a lot of our resources aligned, We presented more updates at our NACOM meeting, which is our internal annual meeting, and then also education for our customers, which is what we're presenting today. And we're very excited to be here to do so. In order for our task force to fully understand the gaps that needed to be filled, we had to kind of take a step back and assess multiple processes. So we'll turn our attention to one of these general processes and then also provide some transparency on some bioMérieux specific processes. So when we take a look at the big picture with CLSI, so the starting point for updating breakpoints is those pieces of information that we recently talked about, where whether it's changing resistance in these organisms, updates in pharmacokinetic or pharmacodynamic information, or clinical outcomes that we see in our literature. So because we have that change that's constantly happening, CLSI is continuously monitoring these updates. They have a subcommittee that reviews this information as it comes in. Sometimes there's a focus on certain bug-drug combinations, and the working group will assess those and then move on to the next, and sometimes they're happening simultaneously. If a decision is made to update a breakpoint, they annually present this in their M100 document. The M100 document is an amazing tool when it comes to breakpoints and the historic component, as well as the most up-to-date breakpoints. They also usually have a webinar, which they had earlier this month, to present those changes. Within that M100 document, you can find the two different tables. One is a review of changes for that current year, and then another is a table that presents the changes since 2010. So with all of these changes, we have to understand now, if CLSI makes an update, what happens next? So this, you can see that in this process, there's a point where there's a new drug, but then there's also FDA, or there's also updates to breakpoints. So in that second column, anytime CLSI decides to make an update, they're first going to publish it in their M100, as I mentioned, and then they're also going to submit this to the FDA. The FDA reviews their rationale document, which includes all the data that they reviewed and all the reasons they believe that the breakpoint should be changed. If it's accepted by the FDA, they then publish that on their STIC, or Susceptibility Testing Interpretive Criteria website. At this point, this is where manufacturers such as bioMérieux, or on here the CASTMAN, the Commercial Antimicrobial Susceptibility Testing Manufacturers, where we can start our process for updating breakpoints. That does take some time, and understanding that even after that's done, we have to submit to the FDA and then get their approval. So we have three moving targets: that of CLSI, that of FDA, and that of the commercial companies such as ours. Excuse me. Excuse me. So within all of these steps, there is an opportunity for customers and their labs to perform a verification or a validation. And this is where a lot of questions came up from our customers. We'll get to the point of prompting this change, but certainly understanding that throughout this process, where there is misalignment between CLSI and the FDA, or the clinical or the commercial manufacturers such as bioMérieux, the labs can decide to perform a validation in order to get up to date with CLSI before the information is available or the updates are available from FDA. That being said, I did want to share the work that bioMérieux has done since 2017. This is a list of all the antimicrobials that have been updated, and they're indicated by their gram-positive or negative, or if they're with a yeast or used against yeast, such as the fluconazole and the caspa. I think a great example is the most recent update from 2023, cefazolin. So up until recently, the labs were only able to use the cefazolin breakpoint for urine on our cards. And this year we did get the approval for the lower breakpoint for systemic use of cefazolin, and that will be on all of our new cards that will be coming out in the fall. And we are very excited for that transition and the hope that there is ease brought to our labs because of the new formulations and breakpoints that will be present on those cards in the fall. So that brings us into some of the resources that bioMérieux has put together in terms of breakpoints. So this is kind of a summary of what bioMérieux has decided to put together. So again, we felt a call to action as a company, as a supplier of one of the automated susceptibility tests used in many labs across the US and the world. So there's quite a few resources. For those of you familiar with bioMérieux University, there will be many on there. We'll also have a dedicated microsite, and I'm really excited about this because it's going to be a one-stop shop for all the materials that we're going to go through, and a place to actually put in requests if you have questions or need assistance with any of this. We'll go through the verification and validation protocols, guidance documents on how to document your baseline breakpoints in use. We'll also have links to some of those external resources you may need, such as the CLSI M100 and the FDA STIC website, and then also frequently asked questions. So this is our tool that we developed, the Breakpoint Audit Tool, abbreviated BAT, and it's just a very small screenshot. And what it's going to allow our customers to do is to document their current breakpoints in use, which is one of the new CAP requirements. It includes CLSI M100 data for that organism antimicrobial combination, the susceptibility breakpoints that are listed and the year that they were approved. It'll also include the FDA website information. So it'll indicate if the FDA site matches those breakpoints presented by CLSI, and if not, it will list the breakpoints that the FDA has approved. It also has information from the instructions for use, or the IFU for your card, and also from the AST selection guide, which we have some visuals of that a little bit later. And that's really important for the drug code. As we continue to update our antimicrobials and get new approvals from the FDA, they do have different drug codes if we update the formulation, so we can allow our customers to implement those breakpoints. There is some information that you'll get from your AES breakpoint report. And this is, the breakpoints that you may have implemented in the change report as well. And then there's the final column there is one that includes the action that you would need to take, verification, validation, or if you're up to date based on the information that is in the card. This is an example of the Vitech AES report. So there's quite a bit of information in here, but essentially what we'll do is, this is a report that you'll use to understand the current breakpoints that are being used in your system. There's information on here that does pertain to which set you're using. So when you work with your FAS, which I highly recommend for updating your breakpoints, they are a great resource to answer any questions or provide advice on. One of the things that you'll want to do is create two different sets. And the reason we suggest that is you'll be able to toggle between the two. So you'll have one with your current breakpoints that you're able to use on your clinical isolates to produce your MICs and then your breakpoint interpretations. And then you'll have the alternate set that you're performing your verification or validation on. There is a tag here that says which status it is, whether it's active or inactive. And that it's just that good indicator so that you can flip back and forth and continue with patient care, as well as updating your breakpoints. There's different information about where the MICs are coming from. In general, in this one, it says 2017, just for transparency. When our software gets updated, by the time it gets out to the customer, it's usually about 2 years old. And that's just because it takes so long for us to update it in our system and then gain approval from the FDA. But we will be keeping up to date on it as much as possible. And then this is a similar report that you can pull from AES, only this one is documenting the changes that you have made as a lab. So it may include information. So for example, on this top one, you can see that the custom breakpoint that was entered on the top row decreased the susceptible breakpoint from 8 to 1, the intermediate from 16 to 32 to 2, and the resistant one from 64 to 4. And you can see that the comment here was from this particular version of the M100. So knowing that this breakpoint was already updated and validated for the system. So that's how you would use that report as you're putting that information into the breakpoint audit tool. Another piece of information, so when the breakpoint audit tool is dispersed, it will include your card number, which will auto-populate the actual drug code. So as this drug code that I've mentioned, it varies between cards and versions of the drug that is approved by the FDA. So some of our older cards may have more narrow susceptibility ranges. Some of our more recent ones will certainly reflect the updated breakpoints from CLSI and FDA. Our old IFUs, the drug code was pretty general. However, on our newer ones, we've updated it to include this type of code. So it looks a little cryptic, but it is the best indicator for the drug formulation on the specific card that you have. And that information can also be found in the AST selection guide. This is a guide that you can use to help you pick which card is best for your hospital, for your patient population. And this is another document that will be updated in the fall when we have new cards available. And then here's just a zoomed in version to show you that column of the drug code that's on that AST selection guide. Next, we have a view in Vitech 2. It's another resource to go look into and understand your current Vitech 2 cards and what is in the system. So you'll log in to your Vitech 2 via the icon, select the tools icon, select the maintain AST card definitions icon, and then you'll pick the card that you want to use. And then displayed there, you have the antimicrobial, the drug code, which wells it's in, which not incredibly important for the purposes of these breakpoints, and then also the calling range that you have for that card. This could become really important if you're at the point and you want to see, do I need to actually change the card that I'm using? You can go in and see what breakpoint range is available on your current card. And if the updated breakpoints are within that range, you likely won't have to switch your card. But if say in the case of Cefazolin a few years ago, when that update came out, most cards did not have that breakpoint low enough, and you wouldn't be able to verify or validate it on your card based on the situation. I'm really excited about the next two slides and the information that we'll share here. So we put together a frequently asked questions document. And this stemmed from some of the questions we were getting in our call center that were coming in through our field application specialists and through our medical information requests in our medical affairs department. So we decided to put together this FAQ document, and it's intended to be used by both our customers and our internal teams because this is such a complex topic. So this is broken down into 6 different sections. The first is general questions. So what is a breakpoint? Why does CLSI decide to update breakpoints? We then move into a section about the process of updating breakpoints. And this is really focused on, should I verify? Should I validate? What's the difference? How do I know when to perform either? And then we also, on the next slide, we'll talk about the resources that we actually have for verification and validation. The third section is what steps bioMérieux is taking, which is certainly a lot of the information that we're covering on the webinars today. The fourth is resources from the FDA and CLSI. These are really essential. These really are to the two bodies that are governing and really putting a lot of great information out there. And they're really dictating what we can do as a company and how we can help you through updating your breakpoints process. The fifth is updating breakpoints in the context of bioMérieux instruments and software. And I can't emphasize enough how much we are here to support labs that are going through this process. Again, we recognize the call to action and the need to be able to update these from a patient safety and public health perspective. So we have, again, these resources are out there, but we have multiple ways for you to contact us. And then lastly, we have a segment on susceptible dose dependent. So as labs are deciding to update breakpoints, including susceptible dose dependent is an option. We do have a presentation later today on susceptible dose dependent and some of the clinical implications of updating breakpoints, but just some Q&A about how to be, or what those implications could look like and the conversations that you may want to be having with your clinicians. So the verification and validation documents, we just created these and started dispersing them to customers a couple weeks ago. We really heard the need for this loud and clear. So there are some labs that have protocols like this already in place. And there are others that said, I don't even know where to start. And we were very happy to provide an outline for this procedure for customers to adapt to be their own. All of this is based on current guidance from CLSI and other agencies that are at the forefront of laboratory procedures. And within that, you'll find a pretty extensive list of the resources and references. We have an introduction within the document, an introduction and kind of how you might want to be using the tool. There are worksheets to record your actual verification and validation and all the steps that you'll take to get to that point. There's guidance that's provided on accuracy, reproducibility, and QC. We also have definitions and calculations. I think it's an entire page actually. And those are related to things like categorical and essential agreement, minor, major, and very major errors, and how to get to that point. So we're really excited for that page to provide some clarity. You'll have a final page with the summary of results, and there's even a spot, if you so choose to use this procedure fully, for the lab director to sign off on the procedure. And as I mentioned, there's a whole page of different resources in there, all of them public resources for you to use as well. So now moving into some of the external resources. So this was a publication by Humphreys, Dr. Abbott, and Janet Hindler, and this was a really great publication that they put out about like kind of even where to start. They cover a lot. I think one of the things that always stuck out to me was like, even what order do I do this in? What are the most important breakpoints that I need to update if I've done nothing since 2010? So a ton of great information in there and a great primer if this is one of the first times your lab is going through this, or if you simply just need a refresher. A few more publications. So I mentioned the one earlier, Raising the Bar by Simner and colleagues. This was the one where there was a survey sent out, and it was voluntary in the 2019 CAP survey, where labs were responding to and indicating which breakpoints they were using that were obsolete or that were up to date. So a great read. It shows a lot of the reasons that they were not up to date and what they were planning to do. So certainly a great call, our call to action here at bioMérieux. This second one, Emerging Resistance, New Antimicrobial Agents, but No Tests. Another great publication by Drs. Humphrey and Hindler. They are so deeply involved in CLSI. I just can't get enough of the content that they put out. So I selfishly love this paper as well as it relates to breakpoints and susceptibility. And lastly, this is kind of in the format of a blog, but an amazing resource that Dr. Prinzi put together. It's on ASM's website, and it really is just a primer and the high-level information. Great place to start if you are new to updating breakpoints. CLSI also puts out a lot of webinars. Their website is full of so much great information, but I get a lot out of their webinars, and I'm constantly going back to them. This one was from June of last year, all about updating breakpoints, and I respect and appreciate the transparency that was provided by this working group in order to provide some guidance to labs and how they can approach this. As I said, there was another webinar. So I think some of them may have a small fee if you don't have a membership with them, but this one from June in particular was free. The one that was earlier this month that I mentioned, they always have that update of the M100 and any changes that happen. So another great resource to reference. M100 I've mentioned a lot. If you do need direct access, it's on here, but you can also just Google CLSI M100 and it'll pop up. The free one is not in PDF form, but 100% usable. You can also purchase the PDF or the book copy. In there, this is one of the tables that I mentioned. This is the update since 2010. If you haven't made updates, or if you're kind of in the middle of making updates, or say you haven't done any since 2018, it's a good place to go in and maybe get a feel for which ones you still need to update based on the year that that CLSI changed the breakpoint. And lastly, the FDA website. So the STIC website, I'm very grateful that they have this. I believe it was a result of the 21st Century Cures Act. So remember that labs, you have the option to go with CLSI or FDA breakpoints. We, as a commercial manufacturer of AST devices here by Tech2, we have to follow the FDA. They are our governing body. So we will always be closest in alignment with them, though our susceptibility ranges on our card may allow you to be better aligned with CLSI. So just keep that in mind. Reminder that CLSI works very closely with the FDA. They are always submitting their rationale documents to them once they update a breakpoint, so they can share that information, and hopefully the FDA will recognize updated breakpoints. I think a recent example where FDA did not recognize was the recent updates in the CLSI M100 33rd edition. There were updates to the aminoglycosides for Enterobacterales, Pseudomonas, and Acinetobacter. And the FDA chose to not recognize some of those updates. So just be aware of that and the differences that are there. And the STIC website does a great job of explaining exceptions. Sometimes it's under the table that it'll say, these organisms don't have current susceptibilities or are not currently recognized from CLSI. And that is all I have for you today. Again, I'm very proud that bioMérieux has taken a stance to support our customers through this. There are multiple ways that you can reach out to us. The easiest might be in the chat here. Any questions that you put in the chat for the webinar will come directly to me, and I will respond via email. The team members at bioMérieux, be it some of your sales folks or your field application specialists, would be a really, really great resources to reach out to. We also have our call center, and behind a lot of this too, if you have off-label questions, if your FAS is not able to help you with that, we have our medical affairs team here to support you. So we hope that the updates and breakpoints will continue to allow us to stay in this fight against AMR, and we want to be there with you every step of the way. Thank you for your time.